Welcome to another session of our evolution seminar series. Today, we're joined by Zach Farrand from the Department of Entomology here at UW-Madison, who's come to share some research on a particularly adorable member of our North American mountains. So let's all give Zach a warm welcome.
All right. Thanks so much for coming, everyone. Could everyone in the back hear me okay? Okay, great. So yes, I'm now in the entomology department working with a group of ground beetles, but I'll be talking about some work from my master's research when I was at Northern Michigan University, where I was studying American pikas. So first off, what is a pika? You would be certainly forgiven if you weren't familiar with what pikas are, being in the Midwest. They don't occur here in Wisconsin. So pikas are a member of the lagomorph order, which means that they're more closely related to rabbits and hares than they are to rodents, even if they look kind of rodent-like. There's only one extant genus in the family Okatonidae, Okatona, and there are 30 species in this genus. Most of the species diversity is in Asia, and we have 2 species here in North America. In Alaska, Northwest Territories of Canada, we have the collared pika, and then in the United States, plus a little bit into Canada, we have the American pika, which is the focus of my study. So a little more background about kind of the family of Okatonidae. So there's one extant genus today, but there used to be many more. But the one genus that we have today, its origins are likely on the Qinghai Tibetan Plateau, whenever the plateau was actually rising. And it's believed that they were getting kind of pre-adapted to cold environments, kind of being in the kind of early stages of the mountains there. And during a global cooling event during the mid to late Miocene, other kind of sister genera of pikas were declining, and this one genus, Okatona, was doing very well, and it spread out across off of the plateau and over into North America, across Beringia, whenever Asia and North America were connected, and that gave rise to the species that we have today. So just like their origins around the Himalayan mountains, today they still have a very montane distribution. By and large, the species are either in kind of high latitude tundra or up in the mountains because they're very cold adapted and very heat intolerant. So if you want to find American pikas here in the US, go into the mountains, specifically around kind of rocky talus slopes. So just so we're all kind of on the same page here, I thought I would play a little video clip of a pika. So if you're hiking around the mountains, wanting to find pikas, a lot of times you'll hear them before you see them. And on cue, they have the most adorable little call. You can hear a response in the background. All right, so now that I've hopefully won over your hearts of why we should care about pikas, say you want to go find some pikas. So say we go out to Colorado. This is actually a photo of somewhere that I was hiking a couple years ago, and I could hear pikas in the distance. So where would we find them? Find them on talus slopes. So these are kind of rocky piles that kind of break off of cliffs above, and pikas can kind of be perched on top of the rocks, but they also go deep within. And these talus slopes provide a lot of habitat structure. So here's some data showing how temperature can fluctuate dramatically on the surface of the talus, but when you go kind of deep within the rocky slopes, it's a really stable, cool temperature. So they're very heat intolerant, so being able to regulate kind of their microclimate is really important for them, and kind of why they're so dependent on talus slopes. So If you have heard about pikas up to now, it's probably in the context of conservation, specifically impacts from climate change. They make a really nice flagship species for trying to understand the impacts of climate change on montane systems. So just to kind of get us all up to speed on this, I thought I'd give us some history on their conservation status. So in 2003, there was a seminal paper identifying patterns of extirpations in a particular part of their range in the Great Basin region. So this was kind of sounding alarm bells that climate change could be really affecting this species. And in 2007, the Center for Biological Diversity petitioned for endangered status at both the federal and state levels. In 2010, this was denied at the federal level, citing things like the patterns of extirpations that had been observed weren't enough to convey that there's a risk at the species at large, and kind of more reasons beyond that, but leave it at that for now. So, but despite this denied protections, there's kind of a steady drumbeat of studies coming out talking about how they're still continuing to find patterns of extirpation. In the Great Basin, 2011, they identified a five-fold increase in extinction rate of Great Basin pikas and upslope range retractions. So as it's getting warmer in the lower elevations, they're moving up in response to that. Still, 2013, California denies protections. And studies just continue to come out during this period talking about identifying patterns of extirpations, a lot of climatic modeling. But really in the last 10 years or so, there have been some studies surfacing trying to place patterns of extirpations into some context. And this study in 2018 was conveying that despite some kind of particular losses, by and large, Great Basin pikas are persisting. And some have gone so far as to say that low elevation losses could be just at historic range margins. And maybe they've gone through patterns of kind of extirpation and recolonization throughout their evolutionary history. But I will say that trying to kind of disregard the impacts of climate change on the species by just compartmentalizing what's going on with particular populations is very controversial. There's a lot of disagreement, and I'd say that by and large, most pika biologists agree that they are certainly under some risk to climate change, and this can vary a lot across the landscape. But it's really trying to understand how the patterns of extirpations we've observed could relate to the species at large. And kind of one technique or tactic we could use to kind of approach this question of kind of what do we do with the knowledge of these extirpations is identifying conservation units. So this is really getting at how do we identify meaningful management units below the species level. I have a quote up there from Meyer illustrating how, describing evolutionary novelty below the species level, it could be just as challenging as identifying species, which can be quite fraught, as many of you know, I'm sure. But there's a few approaches we can take here. So one approach is describing genetic diversity. So we know, if we're losing certain populations, do those represent really unique genetic units? And there's been a good amount of work on this. Here's a publication from one of my co-advisors where he identified 5 major genetic lineages across their range that are kind of roughly based on mountain range distributions. We could look at phenotypic variation, and there's certainly been some work done on that, including work kind of distinguishing pikas regionally by patterns of their calls. So this is looking at duration and frequency of their call patterns. And kind of some of this work combined dropped the staggering 36 subspecies that used to be described for American pikas down to 5, a lot more manageable. And finally, and what I'm most interested in is looking at adaptive variation below the species level. So essentially, if we're losing particular populations, is this having a dramatic effect on the species at large? Like, are we losing kind of unique adaptive alleles as we lose populations? And kind of up to the point when my study had started, there had mostly been some work identifying interspecific adaptive variations. So kind of looking across those 30 pika species. There had been some variation described. So here is a nice study in 2018 looking at elevational distributions of between pika species and looking at adaptations in mitochondrial genes related to that environmental variation. So kind of where my work comes in here is trying to describe intra-specific adaptive variation, so below the species level, and kind of how that can be used to better understand conservation units. So to know what sorts of adaptive traits could vary below the species level, we have to know some things about their biology and ecology, what sort of traits might vary. So let's start with their diet. So pikas, even though they live in relatively unproductive habitats where it looks like this during a really significant part of the year, they don't hibernate, even though their neighbors, the marmots, which are rodents, certainly hibernate. No lagomorph is known to hibernate, so even if it is beneficial, as far as I know, physiologically not capable of doing so. So how they get around this is they build up hay piles. So over the course of the productive summer months, they're out being busy foragers and building up these big hay caches so that they can survive on these plants over the winter. And there's been some really interesting science looking at some of the biochemical properties of the plants that they're selecting. And it's been identified that they seem to be selectively foraging for plants that are high in toxic secondary metabolites, like this alpine avens, which is very rich in tannins. So these are toxic to herbivores, and they do break down over time, becoming more palatable. But the real function of this in a hay pile is it reduces microbial decay over the winter. So yeah, I kind of had this question of, could there be variation in the amount of toxins that pikas are exposed to? So can we find evidence of variation and adaptation regarding detoxification? Next up, I've kind of alluded to this already, but they have a really unique thermal niche. So pikas, they're running real hot. They're really warm little guys. So they have a high resting body temperature of around 104, 105 degrees, but the lethal body temperature is around 109 degrees. So they're already kind of on a knife's edge of living near their upper body limit. And some work has described how their upper lethal limit for being exposed to ambient temperatures is around 2 hours, around 28 degrees Celsius. That's 82 degrees Fahrenheit. So they really don't do well with heat and really rely on those microclimates and talus structure to regulate their body temperature. So, how do they stay so warm? They live in very cold environments, obviously very adapted for this. And there's a lot going on here. So for one, they have really thick coats, very insulating. Also, they have that kind of ideal surface area to volume ratio. So when they want to, they can be very tennis ball shaped, which is just that ideal kind of surface area to volume ratio for retaining heat. They have high resting metabolic rates, like I've already established. And what I'm most interested in is non-shivering thermogenesis. So this takes place in brown fat cells, which have really high mitochondrial densities. And, think back to our intro bio classes. Mitochondria is the powerhouse of the cell where ATP is being produced during oxidative phosphorylation. And essentially, there's an alternative pathway here where the hydrogen protons that pass through the inner mitochondrial membrane producing ATP, they can be uncoupled by these uncoupling proteins. And instead of producing ATP, that potential energy is released as heat instead. And maybe you can see where this is going with how oxidative phosphorylation is very oxygen dependent, and being up in the mountains, sometimes this can be oxygen poor environments. So kind of co-opting the cellular pathway for producing ATP to also produce heat could be a real challenge here. And so the next trait I want to talk about is hypoxia resistance. So, being in Wisconsin, relatively low elevation, because if you go up to the mountains in Colorado, might have a little trouble getting enough oxygen, strained breathing. So what's going on here is not necessarily a change in the oxygen concentration, but as elevation goes up, air pressure goes down, so the atmosphere is just getting thinner. So that's what's really stressing our respiratory systems. And a way that pikas get around this, one study has identified that they could have increased hemoglobin oxygen affinity. So essentially a physiological mechanism to help with living in kind of oxygen poor environments. Other studies have suggested signs of natural selection on mitochondrial genes that could maybe increase efficiency of oxidative phosphorylation. But others have suggested that this could come with a trade-off. So for pikas living in cold environments, all of them basically, so they need to be producing heat in their brown fat cells, and kind of an inefficient oxidative phosphorylation pathway could be helping to produce heat, which would be at odds with the need for higher efficiency in hypoxia-rich environment or in hypoxic environments. So you can see how there's kind of like a trade-off here. And what makes this really interesting for looking at below the species level in American pikas is there's some strong environmental heterogeneity here regarding elevation. So this is a map showing the samples that I had in my study, where they were collected. And the shaded regions correspond with different subspecies. So we have our 5 subspecies. For simplicity, I have them labeled based on kind of major mountain range that they're associated with or geographic region. And the size of the points here relates to the elevation of those study sites. So you can see how in the Pacific Northwest, it's quite low elevation, whereas in the more southerly latitudes, it's quite high elevation. And this just has a lot to do with kind of climatic conditions across the continent, where it gets warmer the further down south you go. So they need to be up higher in the mountains to find that ideal thermal niche. So, excuse me. This brings me to my research questions. First off, the broad question here is, does environmental heterogeneity influence adaptive evolution in American pikas below the species level? So especially things like elevation, but I also have this interest in variation in how extreme the winters get. In my sub-questions, first looking at nuclear DNA. So can we find environmental associations with allele frequencies in the nuclear genome? And I'm also interested in the mitochondrial DNA, looking at, does oxidative phosphorylation efficiency increase with elevation? So essentially, can we find signs of adaptation, signals of selection in mitochondrial genes that could relate to elevational changes? So to describe my methods here, I started with samples from the 5 American pika subspecies, 160 individuals, plus a representative of Okatona colaris, which is the colored pika up in like Alaska, Northwest Territories. And these were collected by my co-advisor, Kurt Galbraith. And one approach here for my nuclear DNA, I used a approach called target capture. So this is where, and like is shown in this illustration, we're capturing certain candidate genes of interest from the genomes of all my samples. So I identified over 400 nuclear genes that could be related to the phenotypes of hypoxia resistance, thermogenesis, and dietary detoxification. So I identified these through literature searches. Some of them are related to pika studies, some are related to studies with other mammals. And essentially how this works is using the pika reference genome, we had probes designed that are based on the sequences of my candidate genes. These bind to the regions of interest in the DNA samples, and we get rid of all the other DNA that's not attached to those probes, and we're left with our candidate genes, and those get kind of selectively sequenced so we can get really nice high coverage data for just my genes of interest. The other approach here is what's called genome skimming. So this is based on the principle that organellular DNA, like mitochondria, is going to be much more abundant in cells than the nuclear DNA is. So just by sequencing kind of all the DNA in my samples, kind of low coverage whole genome sequencing, I should be getting enough mitochondrial reads showing up in just kind of that kind of relatively random sequencing that should have enough to build some nice gene sets here of the mitochondrial genome. So how I process this, the nuclear DNAs, I built a SNP data set. So these are single nucleotide polymorphisms. So most of the genome is going to be the same between my samples. And I was interested in identifying particular sites which represent mutations, and then kind of used that data set for my analysis. On the mitochondrial side, I generated gene alignments for the 13 mitochondrial protein coding genes. And for analysis, so I have two data sets here, a mitochondrial data set and a nuclear data set. Starting with the nuclear, I used this to identify population structure. So I did a principal components analysis, which gave me kind of general genetic structure and then kind of genetic distance between samples. And then I did an analysis called SNMF, which identifies ancestral populations. And for my tests looking for natural selection, I did one group of tests that's kind of categorized as genotype environment association analyses, and another test which is kind of an outlier allele approach, which does not use environmental data and just looks for kind of significant variations based on population structure. On the mitochondrial DNA side, I did a variety of tests that are kind of under the umbrella of this software package called HiFi, which stands for Hypothesis Testing with Phylogenies. And these are mostly based on the principle of non-synonymous versus synonymous substitutions. So a synonymous substitution is when in a protein coding region, there's a change in base and it does not change the amino acid. Non-synonymous substitution is when there's a change in base that does change the amino acid. So it's a nice way to identify kind of signs of natural selection if there's more non-synonymous substitutions, so more changes going on. And then the other analysis I did with mitochondrial DNA is called TreeSap, which uses, it kind of takes the non-synonymous substitutions a step further and looks for changes in kind of biochemical properties of proteins to get an idea of kind of signals of selection via really changes to the biochemistry of certain proteins. So leave it at that for now. It should make more sense as we go through the results. So we'll start with nuclear DNA, noted by our cell nucleus there, and starting with population structure. So kind of what this looks like as I do the analysis, I start with my SNP matrix. So I had over 17,000 SNPs and 120 individuals. And it looks, kind of something roughly like this, where I have like individuals, and then for each SNP, essentially what their genotype is. So these are biallelic SNPs, so two alleles, and kind of what their genotypic composition is. And then I performed a principal components analysis, which gave me genetic distance between individuals, and it looks like this. So by and large, mostly separating individuals out by subspecies. But one of the really interesting patterns here is going on with the Northern Rocky Mountains lineage. So in the principal components analysis, these or the Northern Rocky Mountain subspecies is kind of split up on axis 2. And this is verified with my SNMF analysis, which identified 6 ancestral populations. So if you remember back, there's 5 subspecies, but this analysis was really pulling apart the Northern Rocky Mountains into 2 kind of distinct lineages. And it's worth noting that this has kind of a strong isolation by distance effect, where as you move further away from the boundary of these 2 lineages, the software had a much easier time placing like samples up here into this particular lineage, and then samples like down here into that particular lineage. But closer to the boundary, essentially it gives you ancestry statistics, and those were a little bit closer to one another in trying to decipher these 2 lineages. So obviously a more recent divergence than the other lineages, but it's worth noting that this was verified by a 2024 study, which identified also 6 lineages, which were the same as what I identified. And they used a different sort of genetic approach using GD-RAD-seq. All right, so on to signs of natural selection. So I'll start with a genotype environment test called redundancy analysis, and it's got some nice visuals to kind of walk us through what's going on here. So I start with my SNP matrix, and I have environmental data for all of my samples, which included elevation and mean coldest month temperature, which is a nice gauge of how extreme their winters get. The software performs a linear regression on the SNP matrix with like essentially the relationship of each individual's SNP data with the environment. So, it could look something like this, where if each of these dots is a sample, they have particular environmental variables and genotypes down here. So you can see that like at SNP1, pretty strong relationship with elevation, but kind of a weaker relationship with cold temperature. And then finally, we get this fitted SNP matrix, where essentially all of the SNPs in my data set kind of have these associations with the environmental variables. And then that gets kind of reduced into a principal components analysis, which looks like that. So what's going on here is in the middle, we have our SNPs. So that's like 17,000 data points there in the middle. They all kind of overlap with each other in this plot. And our two environmental predictors, so elevation, mean coldest month temperature, and then averages for each sample in my data set colored by their lineage. So we can see that Southern Rocky Mountains, Central Utah have a positive relationship with elevation. Northern Rocky Mountains, Southeast had a negative association with cold temperature. So that's interesting to look at, but what I'm really looking for here are particular SNPs that could be under selection and really trying to identify the genes that are represented by those SNPs. So this next plot kind of zooms in on these SNPs in the middle, and we can see that there were 25 significant SNPs, 24 of which were most related to elevation, one most related to cold temperature. And this is a nice plot for visualizing this, because you can see that it's essentially the environmental predictors are kind of pulling these SNPs away from the rest of them because they're so closely associated with those environmental variables. So essentially, I just applied a standard deviation to find SNPs that are kind of on the outlier distributions here due to their environmental associations. So these 25 SNPs represented 25 genes. And what I mean by that is, if we have a gene segment here, boundaries denoted by these marks, and then there's a promoter region upstream of the gene that can influence how that gene is expressed. So I said that a SNP was associated with a gene if it's within those gene boundaries or up to 1,000 base pairs upstream. So it could be in a promoter region. So either way, it could be influencing the gene in some way due to its environmental association. So yeah, in this case, I would have two SNPs representing this gene. But in this case, each of the 25 SNPs represented as separate gene. So my takeaway here, redundancy analysis identified candidate loci under selection in response to cold and elevation. So onto the next nuclear tests. So my next model was latent factor mixed models, kind of similar to redundancy analysis in that it's also a genotype environment association test, but it has some different statistics that it uses. And what I did here is I used the SNP matrix like before, and then I used the combination of elevation and cold temperature. So I kind of combined these into an eigenvector using a principal components analysis, because I wanted to see kind of the interaction of these two variables and how that influences signs of selection here. And it identified 34 significant SNPs, which represented 29 genes. And I was curious to see kind of what does the allelic distribution look like across my samples. So I performed a principal components analysis just on these 34 SNPs. And we can see here that essentially one population in the Sierra Nevada Great Basin lineage had most of the kind of allelic differences here. So at most of these sites, these are private alleles only found in this one population, which is found actually in Utah. And essentially most of the other lineages had the same allele, which differed from that one population. And then my environmental agnostic test Bayescan. So this is just that outlier approach based on kind of significant population differentiation patterns. So this gave me 35 significant SNPs representing 34 genes, did a PCA of those SNPs. And we can see that the major distribution here is the Northern Rocky Mountains lineages were mostly different from the rest of them. There is some separation on axis 2 with the central Utah lineage, but kind of hard to see. But axis 1 explains 87% of the data. Axis 2 only explains about 8%. So most of the allelic differences here are between Northern Rocky Mountains and the rest of the lineages. So takeaway, LFMM Bayescan identified significant SNPs, which are candidates for being under selection. So those are some kind of fun plots to look at, some cool geographic patterns, but I'm really interested in identifying which genes could be under selection. So starting with 29 genes, 34 genes, 25 genes, it's kind of a lot to work with. And I took this approach of identifying genes that overlapped between tests as a way to, for one, reduce false positives by finding genes that were represented from more than one test, and also just to get a more manageable short list of genes. So I ended up with 12 genes that I'm going to call overlapping genes. I want you all to remember this term. It's going to come back later in the talk. So these 12 genes that overlap between tests are called overlapping genes. Here they are. And I have them broken up here based on kind of adaptive category, based on literature searching. 3 of them could be related to hypoxia. 2 could be related to thermoregulation. We have some related to dietary detoxification, and then some related to high elevation, which is actually from pika studies. So that was kind of the adaptive category that they were placed in, and why I included them in my data set. And I also wanted to know the kind of functional consequence, or essentially like, are these significant SNPs changing something about these genes? So I looked at synonymous versus non-synonymous substitutions. So 2 of these genes had non-synonymous substitutions. So there's actually a change going on in the protein. And then 7 of these genes had SNPs in putative promoter regions, so like 1,000 base pairs upstream. So it could be changing something about the gene expression. And it's worth noting that it's 7 genes with promoter regions, but it's actually 9 SNPs total. So a couple of those represented the same gene. And my takeaway here, these 3 tests identified significant SNPs, and there were functional consequences in 9 of these genes. So yeah, that's kind of, those 9 genes are kind of my strongest candidates for selection. All right, so let's shift gears and look at the mitochondrial DNA now. So starting with the HiFi tests, so hypothesis testing with phylogenies. And HiFi uses, there's a variety of tests that you can use that kind of approach signs of selection with in different ways. So one test is called BUSTED. It's an acronym, doesn't really matter in this case. What matters is that it's looking for gene-wide selection, and then you're also able to look for selection taking place on particular branches in the phylogeny. There's RELAX, also an acronym. It's looking for intensification or relaxation of selection on particular branches. And when I say particular branches, I was isolating kind of all the branches of each lineage here, so I could identify if there were signs of selection on particular lineages that differed from the other samples. And then the last tests are a couple tests that look at site-specific or like codon-specific selection. So it's not gene-wide, it's not particular branches, it's particular codons under selection. And I found a lot of significant results here. So BUSTED identified 3 genes and 3 lineages. Relax identified 8 genes in 5 lineages. Fubar and Meme identified 29 codons from 12 genes. So this really does support previous work on pica mitochondrial DNA where, yeah, it's rich with signs of natural selection, which probably has something to do with the extreme hypoxia regions that they live in. So, that's a lot to get through. And my approach to kind of parsing this down was to kind of identify a couple standout patterns here. So on the gene level, there was one gene that really stood out, especially in these codon-specific tests, ND5. So ND5 had over 30% of the significant codons, so by far the most of any of these genes. And it's also worth noting that the Sierra Nevada Great Basin lineage showed signs of intensification in the relax analysis. And ND5 being a really strong candidate was supported by a 2013 study, which identified haplotypes that varied across an elevational gradient in British Columbia of like one particular population. And then I also looked at the lineage level where in comparing like busted and relax, I was looking for significance where it was the same gene with the same lineage. So essentially like ND3 had significance in busted and relax where one particular lineage showed signs of selection, which was Northern Rocky Mountains Southeast. ND4, same deal, also found signs of selection in both busted and relax. And one lineage was significant in both of those tests, Sierra Nevada Great Basin lineage. So maybe you're seeing a trend that this lineage is showing up a lot. So we'll get into some discussion on that later. But for now, I'll talk about my final analysis here, tree sap. So this is looking at selection on amino acid properties using phylogenetic trees. And essentially, it looks for significance via 32 different amino acid properties. And it's kind of basing the change in these properties on the particular amino acid changes. So one amino acid property really stood out, and that's pK prime. And it stands for the ionization of COOH. It was kind of brought out by the change between two particular amino acids where a change from isoleucine to valine had an increase in pK prime, and then the reverse direction had a decrease in pK prime. So I dug into some literature of what this amino acid property could mean, and some have hypothesized that an increase in pK prime could heighten efficiency of oxidative phosphorylation proteins. It's believed that it could lead to changes in the hydrophilicity of a protein, and it could lead to reactions becoming more product-driven. I'm not a biochemist, so I'll leave it at that before I get myself into trouble. Why I really want to talk about this property is, for one, it showed up by far the most in my data set, looking at my mitochondrial genes, but also it showed up in a 2018 interspecific study among pika species. So this study used tree sap on pika species that varied by elevation, and they found that as elevation distribution of different species went up, the change in pK prime value went up. So essentially, they were really supporting this hypothesis that maybe change in this amino acid property in mitochondrial genes could be related to increasing efficiency related to hypoxia. So this was a nice hypothesis for me to test. And what did I find but the opposite pattern? So as elevation increased with samples in my data set, pK prime went down. And this is really driven strongly by the central Utah and Southern Rocky Mountains samples, which had pretty strong declines in pK prime compared with a computed ancestral value. And yeah, the takeaway here is kind of an unexpected relationship where there is a decline in pK prime with increasing elevation. Yeah, opposite of what I expected to find. So to bring us back to my research questions, so does environmental heterogeneity influence adaptive evolution below the species level? I mean, I think we found some evidence to support that. It's worth noting that these are candidate SNPs. So, to really parse out if there's actually like adaptation going on, we'll need to do some functional verifications, but I certainly found evidence to point to there could be some intraspecific variation here. I supported this with nuclear DNA, environmental associations with allele frequencies. And when we turn to the mitochondrial DNA question of efficiency of oxidative phosphorylation, didn't really support that, at least with the tree sap analysis. So I found lots of signs of selection in mitochondrial genes, so it's certainly not to say that there isn't changes in efficiency, but as far as testing it with that hypothesis of PK prime, didn't really support our original hypothesis there. So I want to turn now to some discussion on conservation units, bring it back to the original topics here. So, an additional question here would be like, how does intraspecific adaptive variation contribute to understanding of conservation units? And I want to bring up three specific patterns in my data set. There's site-specific patterns, lineage-specific patterns, and then interlineage patterns. So we'll start with site-specific patterns. We'll look to the nuclear DNA. And if you'll remember back to that PCA of the significant SNPs from LFMM, we had this one population from Utah. It's at a site called Mud Lake. And I think what's really going on here is that Mud Lake has some of the highest elevations in my data set. Not the highest, but it's pretty high up there. And it also has quite cold winters for being so far south. It's in the bottom 40th percentile of my cold temperature data. So it was that combination that I think was really driving the patterns in LFMM. And to kind of back this up, if you remember back to those overlapping genes, those 12 genes I talked about earlier, remember that there were two non-synonymous substitutions from two genes. So these are both private alleles unique to this population. So some of the strongest signals of selection in my data set were actually unique to this one population in Utah. So this to say that particular sites can be harboring kind of unique adaptive alleles. And in this case, near the Great Basin, it's in kind of a challenging region for them, kind of conservation-wise. So that's really worth pointing out, that kind of some evolutionary novelty taking place and maybe a challenging environment. Second, I found lineage-specific patterns. So we'll turn to the mitochondrial DNA for this. So go back to my standout patterns in the hi-fi tests. ND5, which showed kind of the strongest signals of selection, showed intensification on the Sierra Nevada Great Basin lineage. And at the lineage level, ND4 also had significance with Sierra Nevada Great Basin lineage from two separate tests. So if you remember back to, it was pikas in the Great Basin region that are a real source of concern among pika biologists. And some have suggested that populations in this region could be at kind of habitat extremes or range margins. So it appears that my data could be suggesting that, yeah, if they are in these really challenging environments for them, there seems to be signs of selection to kind of back up that they are adapting to rather unique environments compared to other pikas in other regions of their range. And finally, my favorite pattern to discuss here is interlineage patterns. So remember back to those overlapping genes, those 12 genes, we had 7 genes where that had SNPs in promoter regions. It was 9 total SNPs. So 7 out of those 9 promoter region SNPs actually had the alleles split between different lineages. So it wasn't unique to one lineage over another lineage. So it looks something like this, where we have the 2 alleles colored on red and blue. And, it could be fixed, like it's, blue allele is only found, or Cascade Range only has the blue allele, Sierra Nevada only has the red allele, or it could be mixed where the 2 alleles could be kind of distributed across the lineage. So, but in other words, yeah, these alleles were kind of split up across the genetic units here. And it's a fair question to say kind of what's going on here. I mean, some of these lineages have been isolated for perhaps over 1 million years. How do we still have patterns of like unique adaptive alleles being maintained? So being maintained across lineages. So one hypothesis here is parallel evolution via common ancestry. So these two alleles could be maintained in kind of ancestral populations and then could reach fixation or could be kind of maintained in lineages as we move to the present. There's convergent evolution where there could be independent evolutionary origins where, this allele here and this allele here derived independently in different geographic regions. Or there could be interlineage introgression, so actually passing on genetic material between lineages. So there is some data to support the idea of kind of interlineage introgression. It's been identified mainly in narrow contact zones. So how this may have influenced the species on a kind of a broader scale, kind of yet to be investigated. But a question I'm really interested in that I'll talk about in a couple slides. So to kind of wrap up this conservation discussion, we'll go over some conclusions here. So my analysis suggested that while there are 5 American pika subspecies, there could very well be 6 lineages. And perhaps we should be thinking of the Northern Rocky Mountains subspecies as kind of 2 distinct genetic units. I think that my analyses also suggest to be considering each lineage independently. I mean, kind of like its own independent unit genetically, but also they all have unique challenges. They live in unique environments and they have kind of a variety of adaptive alleles that could be unique to different lineages. So perhaps each lineage should be considered differently in how we think about particular sites and populations. And I'm especially pointing to the Sierra Nevada Great Basin lineage. It really stood out in my analysis as showing signs of selection, and it seems to be the most at risk due to climate change. So perhaps we should be thinking, very fine scale with this lineage of trying to reduce losses of particular populations, because these could be harboring a lot of kind of novel adaptive alleles. And it's also important to keep in mind that lineages weren't totally independent in my selective signal results. So, there could be signs of parallel evolution or could be signs of admixture, but essentially preserving evolutionary novelty across their range. Perhaps those adaptive alleles could be introgressed between lineages in the future and could be important for them adapting to future climate changes. So if you're interested in this, I'll do a shameless plug of my publication that goes over it all. If you want to read more, I'll point you towards that. And just with the last couple minutes here, I want to talk about some future directions that I'm going with these data, if I can ever find the time to do it. So I'm interested in this idea of adaptive introgression. And we'll go back to evolutionary origins of Okatona, the genus I'm studying. Remember, they spread across Beringia into North America, and there's some evidence to support that this didn't happen once, but actually twice. And there's been some really interesting patterns in their endoparasite phylogenetics. So this is a tapeworm taken from a pika small intestine. And this is some work done by my co-advisor, Kurt Galbraith. And I'll just quickly walk us through this phylogeny. So in red, we have Asian pika parasites. In yellow, we have American pika parasites. And we can see that there is a split between Asian and American pikas in the Miocene, and then another split in this lineage in the Pliocene. And this could be suggesting 2 distinct waves of dispersal across Beringia. But there's fossil evidence to support that this first wave was likely outcompeted by the second wave. But the fact that current day American pikas can still show this signature of divergence in their parasite lineages is really interesting. And, there's a lot of potential hypotheses here, but one that I'm really interested in is whether or not the second wave of pika dispersers could have interacted with the first wave, passing on parasites, maybe passing on genetic material. So I'm interested to see if I can find signs of adaptive alleles being passed from the first wave to the second wave. So we'll see where that goes. And yeah, with that, I will acknowledge everyone involved in my study. So my co-authors and advisors, Kurt Galbraith and Kate Teeter, a lot of appreciation for them and all of the support they've given me in this. I want to also thank Jeff Horn, who manages the Williat computer cluster at NMU. Jeff actually passed away last year, so this work is really a dedication to the support that he's provided students, and this certainly would not have been possible without him making high performance computing so accessible at such a small school in remote rural Michigan. And also want to thank some methodology advising, including my advisor here, Sean Scovell, and acknowledge my funding sources. And with that, you can take any questions. Thanks, everyone. Yeah.
Kind of curious about the distribution of the different subspecies across the geography, and you have some of the ranges look to be very, at least on the zoomed out map, to be very close to each other.
Yes.
Are there geographic barriers?
Yeah.
Like, what are the?
There's a map.
Yeah, like for example, the southern of the Rocky Mountains and the northern of the southern Rocky Mountains, blue and red. Like, are there geographic barriers that are preventing kind of organization here, or is it the bigger nature and stuff?
Definitely. I can't speak to the patterns all the way across their range, but I know like one pattern in particular is like Sierra Nevada, Northern Rocky Mountains. It seems like the Snake River probably separates that, and there were likely different glacial refugia for pikas that were in like the Blue Mountains here versus the Northern Rocky Mountains. So yeah, it shows some really interesting patterns of geographic barriers, both like present day and during past glacial cycles. But as far as I know, I think a lot of the separation here is geographic. Maybe also aided with the fact that pikas are very territorial and have pretty short dispersal. Yeah.
Kind of related to that question, I'm curious on the first PCA plot you have with the population structure. Some of there were some like a couple samples that were pretty distant from the rest.
Oh, sorry. One more. Okay.
Yeah, so the green and the purple, there's
Right.
samples that are, you know, as distant from their populations as some of the others from each other. I'm curious if you have insights on
Ooh.
if they're related, if you know, if they're close geographically to other populations, or why those might be so distant from the rest.
Oh man, you got me on this. I'm sure I looked into this like a couple years ago. I cannot recall which individuals those are. That's a really good question. I wish I had an answer for you right now. Yeah, that's a good spot. So sorry. Yeah.
Yeah, I wanted to ask about your skim sequencing for the mitochondria. You're targeting that, and so I mean, clearly your per site depth of coverage is going to be much higher for the mitochondria, but you'll get some nuclear reads,
Right.
of course. Now, on a per individual level, that depth of coverage at a nuclear site wouldn't be enough to do anything with. But I'm just curious, like, if you were to aggregate the reads, the nuclear reads, you know, across like a whole lineage, let's say, like your 5 lineage groups. I'm wondering, like, maybe you get, you know, up to like 8 or 10x per group, such that you could actually do like window level scans for elevated genetic differentiation. I don't know, do you think you'd have that?
Yeah, that's a great question. And it's worth pointing out that with the target capture process, it also gave me a lot of off-target reads too.
That's a good point too, yeah.
So in my final data set, you know, I targeted over 400 genes, but I had over 500 represented with my SNP data set. So and I've talked with people who have done the same sort of target capture protocol, and that's kind of to be expected. And I use like a low pass whole genome sequencing for the mitochondria, but they were suggesting that even just you might get enough off-target reads from the target capture to get that. So, but yeah, I do think that this technique would give you certainly enough like kind of pooled coverage if you're looking across certain populations. Yeah, I think that would certainly be
Between
possible.
those two sources and at kind of the window level.
Right. Yeah, I think so.
Regarding defining these conservation limits and particularly related to attaining or not attaining some kind of conservation status, do you have an idea of what kind of threshold you would need to meet to make that definition? Like if you have a population where there's on average this many SNPs and this percentage of them can be shown to have like this fitness
Right.
measurement on it, but how do you actually quantify that in terms of how it's related to, protecting or not protecting different populations?
It's a great question. And actually a question brought up by one of my reviewers on my paper of like, how do we think about kind of conservation units where, say, with like salmon, we could have multiple conservation units in the same river versus pikas that have been separated sometimes over 1 million years, and just getting like managers the ability to get their heads wrapped around this evolutionary distance. Yeah, I mean, there have been some other studies that have come out looking at kind of adaptive potential of different lineages and populations. That was sort of based on this interesting ranking where they had like 50 or so biologists that all work with pikas kind of rank their adaptability by different sorts of patterns, and kind of had this predicted pattern where the Great Basin lineage really stood out as being under threat. But yeah, as far as establishing a threshold, yeah, it's tough. I mean, it's also something that needs to be really easy to communicate, especially to lawmakers and managers. So I don't really have a great answer, but it's a really good question. Yeah, Sean.
Maybe push you a little bit more on that. Told it to advise someone about translocation of populations going to go extinct. Which population would you translocate and where? Would you be willing to mix together, and why? Like,
yeah.
To me, it really stands out. This mud lake population, super interesting.
Right, and
High, very cold, and unique. If we lost that, where would you put that so that you would actually gain benefit from that translocation?
Yeah, I mean, one thing you'd really have to be careful for is if they are harboring kind of unique adaptive alleles, if you allow them to introgress with another population, you might lose some of those adaptive alleles just via drift. And so that would certainly be something I would be concerned about, is trying to really hone in on kind of how genetically divergent they are from other populations if you're going to allow intermixing. There's also going to be serious questions about habitat suitability. And, if you do go through this pretty extreme effort of translocating samples, are they going to be able to make it long term? And there's been some really interesting work identifying behavioral plasticity in pikas, where, we usually think of them as being on talus slopes, but there's evidence that they can move into forests to escape heat at times, and they seem to illustrate there's a lot of behavioral plasticity, a lot of dietary plasticity across their range. So I think there would just be like certainly a long multivariate approach to trying to keep environmental conditions as close as possible, and also trying to find populations that are as genetically similar as possible. But it's a dense multi-tiered approach for sure.
Maybe one last one.
Okay.
You have some really weird sites with pikas at very low elevation.
Right.
Are they represented here, and is there anything special about them? Like Lava Beds National Monument, things like that.
Right. So we unfortunately don't have some of the really weird outlier populations. Yeah, like Lava Beds National Monument in Idaho has really extreme population that lives in a very hot environment, but they're able to use structure kind of in lava tubes to get around that. There's also a population in the Columbia River Gorge that's near sea level. They also eat an extremely high proportion of moss, which is really bizarre because moss is a very low quality food source. I don't have those populations in my data set, unfortunately. Other people that have done some work on those populations have identified some really cool patterns, especially with their gut microbiomes, with that kind of moss-rich diet. But Yeah, what was really interesting about this data set and made me so excited is that there was this nice elevational gradient kind of going from like the Pacific Northwest down to more southerly latitudes. So yeah, it's a fair question that if I were to do this study in the future, I'd probably want to go out and get some of those more unique populations and analyze those appropriately.
All right, let's all get after the.